This unit provides an overview of types of microorganisms with a focus on bacteria. The discovery of antibiotics is reviewed along with discussion of the cellular targets exploited by these drugs. Microbiology. Microbiology is a broad term that encompasses the study of all different types of microorganisms. But in practice, microbiologists tend to specialize in one of several subfields. For example, bacteriology is the study of bacteria. Mycology is the study of fungi. Protozoology is the study of protozoa. Parasitology is the study of helminths and other parasites. And virology is the study of viruses. Immunology, the study of the immune system, is often included in the study of microbiology because host-pathogen interactions are central to our understanding of infectious disease processes. Microbiologists can also specialize in certain areas of microbiology, such as clinical microbiology, environmental microbiology, or food microbiology. Most microbes are unicellular and small enough that they require a microscope to be seen. However, there are some unicellular microbes that are visible to the naked eye and some multicellular organisms that are microscopic. An object must measure about 100 micrometers, or microns, to be visible without a microscope, but most microorganisms are many times smaller than that. For some perspective, consider that a typical animal cell measures roughly 10 microns across. Bacterial cells are typically about 1 micron, and viruses can be about 10 times smaller than that. Microorganisms are found in each of the 3 domains of life: bacteria, archaea, and eukarya. Microbes within the domains bacteria and archaea are all prokaryotes. Their cells lack a nucleus, whereas microbes in the domain eukarya are eukaryotes. Their cells do have a nucleus. Some microorganisms, such as viruses, do not fall into any of the 3 domains of life. In this section, we'll briefly introduce each of the broad groups of microbes. Microorganisms differ from each other, not only in size, but also in structure, habitat, metabolism, and many other characteristics. While we typically think of microorganisms as being unicellular, there are also many multicellular organisms that are too small to be seen with a microscope. Some microbes, such as viruses, are even acellular, not composed of cells. There are 5 kingdoms within each domain, a microorganism: fungi, algae or protists, protozoa, viruses, and bacteria. The discipline of microbiology evolved from the need of physicians to test and apply the germ theory of disease and from economic concerns related to the spoilage of food and wine. The initial advances in pathogenic bacteriology were derived from the identification and characterization of bacteria associated with specific diseases. During this period, great emphasis was placed on applying Koch's postulates to test proposed cause and effect relationships between bacteria and specific diseases. Today, most bacterial diseases of humans and their etiologic agents have been identified, although important variants continue to evolve and sometimes emerge. For example, Legionnaire's disease, tuberculosis, and toxic shock syndrome. Bacteria are found in nearly every habitat on Earth, including within and on humans. Most bacteria are harmless or helpful, but some are pathogens causing disease in humans and other animals. Bacteria are prokaryotic because their genetic material, DNA, is not housed within a true nucleus. They have a wide range of metabolic capabilities and can grow in a variety of environments using different combinations of nutrients. Some bacteria are photosynthetic, such as oxygenic cyanobacteria and anoxygenic green sulfur and green non-sulfur bacteria. These bacteria use energy derived from sunlight and fix carbon dioxide for growth. Other types of bacteria are non-photosynthetic, obtaining their energy from organic or inorganic compounds in their environment. All bacteria are unicellular organisms that reproduce by binary fission, the process of replicating materials inside the cell, including the DNA, and dividing to produce 2 daughter cells. Most bacteria are capable of independent metabolic existence and growth, but species of Chlamydia and Rickettsia are obligately intracellular organisms. Bacteria have characteristic shapes. The common microscopic morphologies are cocci, round or ellipsoidal cells such as Staphylococcus aureus and Streptococcus, respectively, rods such as Bacillus and Clostridium species, and long filamentous branched cells called spirochetes. The arrangement of cells is also typical of various species or groups of bacteria. Some rods or cocci characteristically grow in chains. Some, such as Staphylococcus aureus, form grape-like clusters of spherical cells. Some round cocci form cube packets. Bacterial cells of other species grow separately. The microscopic appearance is therefore valuable in classification and diagnosis. The higher resolving power of the electron microscope not only magnifies the typical shape of the bacterial cell, but also clearly resolves its prokaryotic organization. Two types of surface appendage can be recognized on certain bacterial species: the flagella, which are organs of locomotion, and pili, Latin hairs, which are also known as fimbriae, Latin fringes. Flagella can be useful in identifying the type of bacteria. For example, they are found on many species of bacilli, but rarely on cocci. The ability of bacteria to swim by means of the propeller-like action of the flagella provides them with the mechanical means to perform chemotaxis, movement in response to attractant and repellent substances in the environment. Response to chemical stimuli involves a sophisticated sensory system of receptors that are located on the cell surface and/or periplasm. The terms pili and fimbriae are usually used interchangeably to describe the thin hair-like appendages on the surface of many gram-negative bacteria, and proteins of pili are referred to as pilins. Pili are more rigid in appearance than flagella. In some organisms, such as Shigella species and E. coli, pili are distributed profusely over the cell surface, with as many as 200 per cell. Gram stain is a common technique used to differentiate two large groups of bacteria based on their different cell wall constituents. The gram stain procedure distinguishes between gram-positive and gram-negative groups by coloring these cells red or violet. Gram-positive bacteria stain violet due to the presence of a thick layer of peptidoglycan in their cell walls, which retains the crystal violet these cells are stained with. Alternatively, gram-negative bacteria stain red or pink, which is attributed to a thinner peptidoglycan wall that does not retain the stain. Gram-negative bacteria have a cell wall that is composed of the peptidoglycan layer, as well as an outer membrane containing lipopolysaccharides, or LPS, that covers the peptidoglycans. Gram staining involves three processes: staining with a water-soluble dye called crystal violet, decolorization, and counterstaining. Due to differences in the thickness of a peptidoglycan layer in the cell membrane between gram-positive and gram-negative bacteria, gram-positive bacteria with a thicker peptidoglycan layer retain crystal violet stain during the decolorization process, while gram-negative bacteria lose the crystal violet stain and are instead stained by the safranin in the final staining process. The process involves three steps. Cells are stained with crystal violet dye. Next, a Gram's iodine solution, iodine and potassium iodide, is added to form a complex between the crystal violet and iodine. This complex is a larger molecule than the original crystal violet stain. A decolorizer, such as alcohol or acetone, is added to the sample, which dehydrates the peptidoglycan layer, shrinking and tightening it. The large crystal violet iodine complex is not able to penetrate this tightened peptidoglycan layer and is thus trapped in the cell in gram-positive bacteria. The thinner peptidoglycan layer of gram-negative cells is unable to retain the crystal violet iodine complex, and the color is lost. A counterstain is added to the sample, staining it red. Since the counterstain is lighter than crystal violet, it does not disrupt the purple coloration in gram-positive cells. However, the decolorized gram-negative cells are stained red. Another way to identify the type of bacteria in the laboratory is through determination of the presence of antigens on the surface of the cell. Cell wall, or LPS, O, flagellar, H, and capsular, K, antigens are used to aid in classifying certain organisms at the species level, to serotype strains of medically important species for epidemiologic purposes, or to identify serotypes of public health importance. Serotyping is also sometimes used to distinguish strains of exceptional virulence or public health importance. What are antibiotics and how do they work? Any substance that inhibits the growth and replication of a bacterium or kills it outright can be called an antibiotic. Antibiotics are a type of antimicrobial designed to target bacterial infections within or on the body. This makes antibiotics subtly different from other main kinds of antimicrobials widely used today. Antiseptics are used to sterilize surfaces of living tissue when the risk of infection is high, such as during surgery. Disinfectants are non-selective antimicrobials, killing a wide range of microorganisms, including bacteria. They are used on non-living surfaces, for example, in hospitals. Of course, bacteria are not the only microbes that can be harmful to us. Fungi and viruses can also be a danger to humans, and they are targeted by antifungals and antivirals, respectively. Only substances that target bacteria are called antibiotics, while the name antimicrobial is an umbrella term for anything that inhibits or kills microbial cells, including antibiotics, antifungals, antivirals, and chemicals such as antiseptics. Most antibiotics used today are produced in laboratories, but they are often based on compounds scientists have found in nature. Some microbes, for example, produce substances specifically to kill other nearby bacteria in order to gain an advantage when competing for food, water, or other limited resources. However, some microbes only produce antibiotics in the laboratory. The term antibiotic was coined by Selman Waxman, who worked at Rutgers University, and together with Albert Schatz and Elizabeth Bugge, discovered the antibiotic streptomycin. Soon, scientists were able to develop synthetic compounds that had antibiotic properties. Antimicrobials are defined as chemicals that kill or inhibit growth of microorganisms. Antimicrobials include three subgroups: antibiotics, which are used to selectively kill or inhibit microorganisms within the body; antiseptics, which are applied on living tissue to prevent infection; and disinfectants, which are used to kill or inhibit microorganisms on non-living surfaces. The terms antibiotic and antimicrobial are often used interchangeably. An exception to the term microorganism is viruses. Antibiotics do not kill viruses, so they cannot be used for the common cold. There are many compounds that are able to kill microorganisms. However, they are not all called antibiotics. To be an ideal antibiotic, it should have the following other properties besides being able to kill microorganisms. 1. Selectivity. The antibiotic must kill or inhibit the infecting microorganism, but cause minimal harm to the host cells. Water solubility. The antibiotic must be soluble in water to a sufficient extent so that it can be transported through body fluids to the infective sites. Some antibiotics are poorly soluble in water. However, some solubility is essential for effectiveness. 3. Few side reactions. Side reactions of the antibiotic should be minimal. These include possible allergic reactions and negative interaction with food or other drugs that the patient may be taking. 4. Stability. This includes shelf stability and biostability. The antibiotic should have a long shelf life to be economically useful. It should preferably be stable at room temperature. However, there are many antibiotics that need to be stored in the refrigerator. Once taken by the patient, the antibiotic should remain unaltered in the body fluids for sufficient time to be able to carry out its function. Foreign molecules in the body will eventually be either degraded, usually in the liver, or excreted with urine. For the ideal antibiotic, both these processes should be slow. However, rapid excretion is usually a desired property for treating urinary tract infections, since a high concentration of the drug is achieved in the urine. 5. Low cost. The cost of manufacturing the antibiotic should be low enough for patients to be able to afford it. 6. Slow resistance development. Microorganisms have developed resistance to most antibiotics. An ideal antibiotic will be the one to which resistance develops at a slow rate. This will depend not only on the characteristics of the antibiotic, but also of its frequency of use. While some antibiotics are chemically synthesized, a majority of the antibiotics that are used today are produced by microorganisms. Soil is the best place to search for antibiotics. It is known that the soil is a very complex ecosystem in which the inhabitants have developed chemical defenses against each other as a response to competition for nutrients. So it was obvious for many scientists to search for these chemicals. Also, even before the discovery of antibiotics, scientists had wondered about another aspect of the soil. In the history of mankind, or animal kind, many people and animals have died because of bacterial infections. By the time someone dies because of infection, the bacteria have multiplied within the body to reach billions in number. When these dead bodies are buried or just left to decay, all those bacteria are released into the soil. So by now, the soil should be full of pathogenic bacteria and should be the most dangerous thing to even touch. But that is not so. So where did all the pathogenic bacteria go? Now we know that there are numerous microorganisms in the soil that produce antibiotics that probably kill the pathogenic bacteria coming from the dead bodies. So if one wants to search for antibiotics, the best place to search is the soil. That is what scientists did. Of all the antibiotics known today, the majority of them have been isolated from microorganisms present in soil. Some examples are terramycin, vancomycin, and streptomycin. Alexander Fleming, working at St. Mary's Hospital in London, is widely credited for the discovery of the first modern antibiotic, penicillin, in 1928, for which he was awarded the Nobel Prize. Fleming noticed that in one of his old plates, petri dishes, left in the laboratory, colonies of the bacteria Staphylococcus, that causes skin diseases, had lysed, probably because of a contaminating greenish mold growing in an adjacent area of the plate. This led to the discovery of penicillin. By extracting the substance from cultures of the mold, he was able to demonstrate its antibacterial activity not only on the plates, but also when given to infected mice. Fleming named this substance that killed bacteria penicillin after the penicillium mold from which it was obtained. Penicillin functions by inhibiting the synthesis of bacterial cell wall. In 1943, Salman Waxman, Albert Schatz, and Elizabeth Bugge at Rutgers University screened many microorganisms from the soil and discovered an antibiotic from the bacteria Streptomyces griseus. They named it streptomycin, which was the second antibiotic obtained from soil bacteria. The first one was gramicidin. Streptomycin was effective against several diseases, but its most important use was in treating tuberculosis that's caused by Mycobacterium tuberculosis. This was the first antibiotic that could cure tuberculosis. One problem with streptomycin was that bacteria developed resistance to the antibiotic at a much faster rate than penicillin. One way to classify antibiotics is based on the targets in the microbial cell that they interact with to cause growth inhibition. Accordingly, there are 6 major classes of antibiotic. 1. Those that inhibit bacterial cell wall synthesis. 2. Those that disrupt the cell membrane. 3. Those that inhibit the synthesis of important metabolites. 4. Those that inhibit DNA synthesis, replication. 5. Those that inhibit RNA synthesis, transcription. And 6. Those that inhibit protein synthesis, translation. Another way to classify antibiotics is based on their effect on growth and survival of the bacteria. Those antibiotics that kill bacteria are called bactericidal, like penicillin, and those that stop the growth of bacteria but do not kill them are called bacteriostatic, like chloramphenicol. Note that bacteriostatic antibiotics can also eventually kill bacterial cells if used at large concentration. Similarly, bactericidal antibiotics can also behave as bacteriostatic if the concentration used is too low. So these two terms are better defined as follows. An antibiotic is bacteriostatic if it inhibits growth of bacteria but does not kill bacteria cells at a safe and practically achievable concentration. An antibiotic is bactericidal if it kills bacterial cells at a safe and practically achievable concentration. If a bacteriostatic agent is removed, then the bacterial growth will resume. So does this mean that bacteriostatic antibiotics are not useful? No, bacteriostatic antibiotics are often prescribed by doctors. This is because by stopping growth of the infecting bacteria, it gives the body's immune system sufficient time to kill the bacteria. So they work along with the help of the immune system. In fact, even bactericidal antibiotics sometimes do not kill 100% of bacteria. But by bringing down the total number of bacteria, they help the immune system tackle the problem. Bacteriostatic antibiotics function by stopping an essential metabolic process, which can later resume when the antibiotic concentration decreases. Bactericidal antibiotics cause irreversible damage to their targets, thereby killing the cells. The learning goals for this unit, Microorganisms and Antibiotics, are as follows. 1. Define microbiology as a scientific discipline. 2. Match types of bacteria with morphologic and laboratory observations. 3. Draw the difference between gram-positive and gram-negative bacteria in the context of cellular anatomy. 4. Describe the major cellular targets of antibiotics. 5. Distinguish between bacteriostatic and bactericidal antibiotics.